It's wild, isn't it? Like we're living in this sea of microscopic life, you know, and these tiny things, these microbes, they can totally wreak havoc on us. It's always made me wonder, how do these little guys actually cause these huge infections? Do we need like I don't know, a PhD in microbiology to really get it?
Well, hopefully not, and that's exactly what we're going to try to break down in this deep dive today. Our mission is to really understand how microbial infection and pathogenesis work, basically,
Yeah.
the nuts and bolts of how these microbes interact with our bodies and make us sick.
Right, so it's all about those first steps, right?
Yeah.
How they get a foothold, how they set up shop, and then all the different things that affect just how bad the infection gets.
Mhm.
It sounds like we're going to need a good map for this journey.
We do, and luckily our source material today is like a detailed guidebook. It covers all the key areas, starting with adherence, that initial latching on process. Then we'll move on to colonization and invasion, how those microbes spread and establish themselves.
And we can't forget about those terms. We always hear pathogenicity and virulence. That's all about their ability to actually cause harm, right?
Absolutely. And then we'll get into the nitty-gritty of their weapons, things like enzymes, exotoxins, and
Oh,
endotoxins.
and of course, we can't overlook how our own defenses or lack thereof play a role in all of this. It's really a fascinating, albeit slightly creepy world when you think about it.
No kidding. All right, so let's start at the very beginning, just like our source material does. Adherence. It seems pretty obvious, but if a microbe wants to cause trouble, it first has to actually stick around, right?
Exactly. They can't just float on by. They need to grab onto our tissues and hang on tight.
So it's like planting a flag on a really, really windy mountain. You've got to secure it first. And where do they tend to do this sticking? It's not just random, is it? Right. It often happens on those surfaces that are exposed to the outside world, like our mucous membranes,
Mhm.
in our respiratory tract or digestive system, and even the surfaces of our teeth. But something that really stood out to me is that adherence can also happen on artificial surfaces, like those found in medical implants.
Oh, wow. Yeah, that makes sense. And that opens up a whole other can of worms, doesn't it? Like in terms of
Absolutely.
complications and infections.
It definitely does. And one of the things we often see with those artificial surfaces is the formation of biofilms. So imagine a community of microbes all huddled together on a surface, and they're encased in this matrix that they produce themselves, almost like like a city of microbes covered in slime, I guess.
Ew. Yeah, I get the picture. And biofilms are particularly problematic because they're incredibly resistant to antibiotics, aren't they?
Yeah. They are. They make infections associated with implants notoriously difficult to treat. And there are a few reasons for that. First, the biofilm itself acts as a physical barrier, making it hard for antibiotics to penetrate and reach the bacteria inside. But then there's also the fact that bacteria within biofilms tend to grow more slowly, and their metabolism changes, which inherently makes them less susceptible to those antibiotics.
So it's like they've built this fortress and they've changed their lifestyle to make them even harder to kill. It's a pretty ingenious defense strategy when you think about it.
It really is. And that's why researchers are always looking for new ways to combat these biofilms.
And our source material actually mentioned a pretty fascinating approach, biomimicry. What's that all about?
Well, it's basically taking inspiration from nature. You know how some surfaces in nature are really good at repelling bacteria, like shark skin, for example, or lotus leaves. They have these tiny structures on their surfaces that prevent bacteria from attaching in the first place. So scientists are trying to mimic those structures using materials like titanium dioxide to create implant surfaces that actually kill bacteria on contact.
Whoa. So it's like building those defenses right into the material. So it's way better than constantly trying to fight off infections that have already taken hold.
Okay.
Okay, so let's zoom in a bit more now. On a molecular level, how does this whole sticking process actually work? Do they, like, have tiny little grappling hooks or something?
Not quite. But it's just as amazing. It all comes down to very specific interactions between molecules on the surface of the pathogen and molecules on our own cells. So pathogens have these things called adhesins, which are often glycoproteins or lipoproteins, and these adhesins act like keys that fit into specific locks, which are the receptor molecules on our cells.
So it's like a lock and key system, and only the right key can open the right lock.
Exactly. And these receptors on our cells can be a variety of things, like glycoproteins or glycolipids. And this whole lock and key thing is what determines which pathogens can bind to which cells or tissues in our body.
Okay, so the specific adhesins a microbe has and the types of receptors we have on our cells will basically determine what that microbe can infect, right?
Mm.
Like whether it goes for the lungs or the intestines or somewhere else entirely.
Exactly. And there's a whole bunch of different bacterial structures that can act as adhesins or help with attachment. Things like capsules, fimbriae, piliae. Even flagella, which are usually thought of as helping bacteria move around, can sometimes play a role in adherence too.
Wow, so they've got a whole toolbox of sticky tools.
Mm.
So let's talk about a real-world example here. The chapter mentions dental plaque. Now, I think we've all experienced that at some point.
Oh yeah, definitely. It's a perfect example. So it starts with bacteria non-specifically sticking to that film that naturally forms on our teeth, the salivary pellicle. And then specific oral streptococcus come along and they start producing these sticky substances like capsular polysaccharides and dextrin.
They're basically making their own glue.
Exactly. And that allows more and more bacteria to stick. And eventually they form that mature dental plaque, which is basically a biofilm, like we talked about earlier. And if we don't brush and floss, that plaque can lead to cavities.
Right, so it's not just some random pileup of bacteria. There's actually a very specific process that starts with that initial adherence. Like that they've managed to stick. But they can't just hang out on the surface forever, can they? What happens next?
Right. So the next big step is colonization. This basically means that the microbe starts growing and multiplying after it's gotten a grip on those tissues. And it's important to remember that this is different from our normal microbiota, those microbes that live on and in us all the time without causing any harm. Pathogens, the ones that cause disease, they need to actually set up a home and multiply if they want to make us sick.
So they're not just visiting, they're moving in and starting a family. And our chapter points out that this often starts at mucous membranes, which, makes sense. But don't those membranes have ways to fight back? Like, what about all that mucus?
That's a good point. Mucus is actually a really important part of our innate immune system. It traps microbes and prevents them from attaching. But some clever pathogens have found ways to get around that. They have special adhesins that allow them to stick directly to the epithelial cells of those mucous membranes, basically bypassing the mucus barrier.
So they've found a way to sneak past security.
Sneaky little buggers. They are. And, this whole colonization thing, it can also lead to the formation of those biofilms we talked about earlier. They're a major player in chronic infections because they're so good at protecting those microbes.
Yeah, those biofilms are like the ultimate hideout. Okay, so we've got adherence and colonization covered,
Yeah.
but sometimes it goes even further than that, right?
Yeah.
Sometimes those microbes decide to actually invade our tissues.
Right. And that's when things can get really serious. Invasion basically means that a pathogen can actually get inside our cells or tissues and then potentially spread to other parts of the body. This is what distinguishes a localized infection, where the microbe stays put in one area, from a systemic infection, where it spreads throughout the body.
So like a skin infection versus something like sepsis, right? Big difference.
Huge difference. And actually, the chapter talks about two important terms related to bacteria in the bloodstream: bacteremia and septicemia. It's important to understand the difference between these two.
Yeah, those always trip me up. Can you break it down for us?
Sure. So bacteremia simply means that there are bacteria in the blood. And this can be a transient thing, meaning they're just passing through, and it doesn't always mean you're going to get sick. Septicemia, on the other hand, that's when the bacteria are actively multiplying in the blood. And that can trigger a whole cascade of dangerous events in the body. It can lead to septic shock, which can be life-threatening.
So bacteremia is like a brief stopover, while septicemia is like they've set up camp and are throwing a raging party in your bloodstream.
That's a good way to put it. And of course, there's also viremia, which is the term for viruses in the bloodstream. So whether it's bacteria or viruses, getting into the blood and spreading throughout the body is a major step in causing serious disease.
Okay, so we've covered how they stick, how they set up shop, and how they might spread. Now let's get to the heart of what makes them harmful: pathogenicity and virulence. These terms always seem to go together, but they have different meanings, don't they?
Absolutely. Pathogenicity is simply the ability of a microbe to cause disease. It's either a yes or a no. Virulence, though, that's more about how good they are at it. It's a measure of how much damage a pathogen can cause.
So, like, a cold virus is pathogenic because it can make us sick, but it's not very virulent because it usually just causes mild symptoms. But something like, say, Ebola is both pathogenic and highly virulent because it can cause very severe disease.
Exactly. And scientists actually quantify virulence using something called LD50.
Oh yeah, LD50, that stands for lethal dose 50, right?
Yep. It's the number of microbes or the amount of toxin that it takes to kill 50% of a group of test animals. So a pathogen with a low LD50 is super virulent because it only takes a few of them to cause a lethal infection. High LD50, on the other hand, means it's less virulent.
So like a microbial supervillain would have a really, really low LD50. Scary stuff. We've talked about some things that contribute to virulence, like the ability to stick and invade, but our source material also talks about these things called virulence factors. What are those exactly?
Virulence factors are basically all the tools and tricks that pathogens use to infect us, evade our defenses, and cause damage. It's a pretty broad category that includes things like adhesins, enzymes that break down tissues, and those toxins we'll be talking about soon. It's really their secret weapon stash.
Wow, so they come prepared. Now, I also remember reading about the attenuation of virulence. Is that basically like making a pathogen less dangerous?
That's exactly it. Attenuation means weakening or even completely eliminating a pathogen's virulence. This can happen naturally through mutations, or scientists can do it in the lab by growing pathogens under specific conditions.
And why would they want to do that?
Well, these weakened or attenuated strains are really important for developing vaccines. They can still trigger an immune response in our bodies so we get protection, but they're much less likely to make us sick. It's like showing our immune system a training dummy before it has to fight the real thing.
So it's like a sneak peek for the immune system.
It's pretty smart.
Okay, so up until now, we've been focusing on the pathogen side of things, but the chapter also emphasizes that the outcome of an infection isn't just about the microbe itself, right? Our own bodies play a huge role.
Oh, absolutely. It's really about the interplay between the pathogen and the host. So, how strong are our defenses? What's our overall health like? Those things can really impact how susceptible we are to infection and how severe the disease will be. For instance, many pathogens carry these virulence genes, and sometimes they can even share those genes with each other. It's like trading tips on how to be a better bad guy.
So they're like little microscopic criminals sharing their secrets.
Kind of, yeah. And sometimes those virulence genes aren't even on the main bacterial chromosome. They can be on these mobile genetic elements like plasmids and something called pathogenicity islands.
Pathogenicity islands. That
Yeah.
sounds ominous. What are those?
Imagine a bacterial chromosome, right? And then you've got this cluster of genes that all work together to make the bacteria more dangerous. That's a pathogenicity island. And bacteria can acquire these islands through a process called horizontal gene transfer, which is essentially swapping genetic material. So they're like sharing weapons blueprints. A good example of this is Salmonella. It uses genes on these pathogenicity islands to cause infection, things like adhesins to help it stick to our intestines, exotoxins to cause inflammation, and even proteins that help it dodge our immune cells.
So it's like they're constantly upgrading their arsenal.
Yeah.
Not good for us.
Mm.
And on the other side of the equation, as you said, there's the host, and the chapter talks about compromised hosts. Who are those exactly?
A compromised host is basically anyone whose immune system or defenses are weakened, making them more vulnerable to infections. This can be due to all sorts of things, like age, other diseases, medical treatments, even lifestyle factors. Like babies and the elderly often have weaker immune systems. And people with conditions like HIV or who are undergoing chemotherapy are also more susceptible to infections.
So their defenses are down, and that makes them an easier target. And this brings us to those terms: opportunistic pathogens and nosocomial infections.
Right. Opportunistic pathogens are those microbes that usually don't cause problems in healthy people but can be really dangerous in compromised hosts. And nosocomial infections are those that people pick up in hospitals or healthcare settings, which are obviously a big concern for compromised patients.
It really highlights the importance of a healthy immune system. Okay, so we've laid the groundwork: adherence, colonization, invasion, pathogenicity, and the interplay between the microbe and the host. Now let's dive into the specific weapons that pathogens use, starting with enzymes.
Enzymes are essential for life, but pathogens have figured out how to use them to their advantage, basically weaponizing them to make us sick. They use them to break down our tissues, get nutrients, and even disarm our defenses.
So they're like little molecular saboteurs. Can you give us some examples of these enzymes in action? The chapter mentions some pretty interesting ones.
Oh yeah, there are some real doozies. Take hyaluronidase, for example. It breaks down hyaluronic acid, which is a major component of the stuff that holds our tissues together. So by breaking that down, pathogens can move more easily between our cells. Then there's collagenase, which targets collagen. Collagen is the most abundant protein in our connective tissues, so breaking that down allows pathogens to penetrate deeper into those tissues.
So they're basically like a microscopic demolition crews breaking down the walls of our bodies.
Exactly. And then you've got streptokinase, which actually dissolves blood clots. This can help pathogens spread from one area to another, since those clots can sometimes wall them off. But then on the flip side, you have coagulase, which actually promotes clot formation.
Wait, why would a pathogen want to create a clot? Doesn't that trap them?
Well, it might seem counterintuitive, but for some pathogens, those clots can actually act as a shield, protecting them from our immune cells. It's like hiding out in a bunker.
Pretty clever. So they can either break down barriers or build their own. And the chapter also mentions enzymes that specifically target our immune defenses.
Right. Some pathogens are real specialists in that area. For example, Enterococcus faecalis can actually modify its own cell wall to make it resistant to lysozyme, which is one of our natural antimicrobial enzymes. And then you have bacteria like Neisseria that produce agases, which break down IgA antibodies. IgA antibodies were really important for protecting our mucous membranes, so by breaking those down, these bacteria can more easily colonize those surfaces.
Wow, it's amazing how they've evolved these highly specific countermeasures to our defenses. It really is an arms race, like you said before.
It is, and it's constantly evolving, which is why infectious disease research is so important. Okay, so we've covered enzymes. Now let's move on to exotoxins. These are a whole other level of nasty.
Yeah, these are the big guns, right? Exotoxins are those really potent poisons that bacteria can produce.
Exactly. They're toxic proteins that are secreted by both gram-positive and gram-negative bacteria, and they can cause a lot of damage even far away from the original infection site. And what's really remarkable is that many exotoxins are incredibly specific in how they work. They target particular receptors or processes within our cells.
Talk about a targeted attack.
Yeah.
And the chapter breaks down exotoxins into three main types based on how they work. AB toxins, cytolytic toxins, and superantigen toxins. Let's start with AB toxins. What's the deal with those A and B subunits? Why do they have two parts?
The A and B subunits each have very specific roles. Think of the B subunit as the delivery truck, and the A subunit is the package it's carrying. The B subunit binds to a specific receptor on the surface of the host cell, and that binding allows the A subunit to get inside the cell where it can do its damage.
So the B subunit gets it to the right address, and then the A subunit unleashes chaos inside.
Got it.
Oh.
What are some examples of these AD toxins and the havoc they wreak? Oh, there's some famous ones. Diphtheria toxin, produced by Corynebacterium diphtheria, is a classic example. It basically shuts down protein synthesis in our cells, which can be fatal. Then you have botulinum toxin from Clostridium botulinum, which you've probably heard of because it's the one that causes botulism.
Yeah, that's the one you have to worry about with improperly canned food, right?
Right. And it causes paralysis by blocking the release of acetylcholine, which is a neurotransmitter that's essential for muscle function. Then there's tetanus toxin, also known as tetanospasmin, which is produced by Clostridium tetani. This one causes the opposite type of paralysis, spastic paralysis, where your muscles contract uncontrollably. It does this by blocking the release of inhibitory neurotransmitters. And finally, we have cholera toxin, produced by Vibrio cholerae, which causes the severe diarrhea that's characteristic of cholera. It does this by messing with ion transport in the intestines, leading to a massive loss of fluids.
Okay, so each one has its own unique way of wreaking havoc, but they all share that two-part structure with the B subunit acting as the delivery system. Pretty sneaky. What about the second type, the cytolytic toxins? I'm guessing those are the ones that literally blow up our cells.
Oh, are that cytolytic toxins, also called cytotoxins or hemolysins, directly damage cell membranes, causing those cells to burst open. They can do this by forming pores in the membrane or by breaking down the lipids that make up the membrane.
So they either poke holes in our cells or dissolve them from the outside in. Yikes. And I remember the chapter mentioning a few specific examples of these membrane destroyers.
Right, so one example is alpha toxin from Clostridium perfringens. It's an enzyme that specifically breaks down lecithin, which is a major component of cell membranes. Streptolysin O, produced by Streptococcus pyogenes, is another one. It forms pores in cell membranes, disrupting the balance of ions and causing the cell to swell and burst. And then you have staphylococcal alpha toxin from Staphylococcus aureus, which also forms pores in cell membranes, leading to cell death.
So they're basically like microscopic assassins, targeting our cells and making them explode. Okay, last but not least, we have the superantigen exotoxins. Those sound like they cause a real mess.
They do. Superantigens are different from the other two types of toxins we talked about. They don't directly kill cells or inhibit specific processes. Instead, they trigger a massive, uncontrolled immune response. They do this by basically short-circuiting the normal way that our immune system recognizes antigens. They bind to both antigen-presenting cells and T cells at the same time, which causes a huge number of T cells to become activated, even if they're not specific for that particular antigen.
So it's like they're pressing the alarm button on our immune system, but they're holding it down continuously.
That's a good analogy. And this massive T cell activation leads to a flood of cytokines, which are signaling molecules that trigger inflammation. This can cause systemic inflammation, fever, a drop in blood pressure, and even shock. Toxic shock syndrome, or TSS, is a classic example of a disease caused by superantigens.
Wow, so in this case, it's our own immune system that ends up causing the damage, thanks to the superantigen pulling the strings. That's pretty terrifying. Okay, so we've covered exotoxins, those secreted proteins that cause all sorts of trouble. But the chapter also discusses endotoxins, and those are a bit different, right?
Right. Endotoxins are not secreted proteins. They're actually part of the structure of gram-negative bacteria. They're lipopolysaccharides, or LPS, that make up a big chunk of the outer membrane of these bacteria. And it's the lipid A portion of LPS that's actually toxic.
So unlike exotoxins, which are actively released by the bacteria, endotoxins are kind of just there, part of their structure. And they get released when the bacteria die and break apart, right?
Exactly. And when that happens, that lipid A can trigger a whole bunch of effects in our bodies. It can cause fever, inflammation, diarrhea, and in severe cases, even endotoxic shock, which can be fatal.
So it's like a final parting shot as they die.
Yeah.
Not very nice. And the chapter mentions that endotoxins are usually less potent than exotoxins, right?
Yeah, in general, you need a lot more endotoxin to cause the same level of effect as an exotoxin, but there's still nothing to mess with.
Definitely not. And finally, the chapter mentions a way to test for the presence of endotoxins, the LAL assay. What's that all about?
The LAL assay is a really sensitive test that uses the blood cells of horseshoe crabs, believe it or not. These amoebocytes, as they're called, have these granules that clot when they come into contact with endotoxins. So it's a really useful way to detect even tiny amounts of endotoxin contamination, which is important for things like pharmaceuticals and medical devices.
That's pretty amazing. Who knew horseshoe crabs could be so helpful in ensuring product safety? Okay, so we have truly taken a deep dive into the world of microbial pathogenesis today. From that initial adherence to the incredible diversity of weapons they have at their disposal, it's been a wild ride. I definitely have a newfound appreciation and maybe a little bit of fear for these microscopic foes.
It really is a complex and constantly evolving field, but understanding how microbial infections work is crucial for developing new ways to fight them. We've covered a lot of ground today, from the different ways that pathogens adhere, colonize, and invade our tissues to the various virulence factors they use to cause harm. We talked about enzymes, exotoxins, and endotoxins, and we even touched on the importance of host factors in determining the severity of an infection. It's all connected, and it all highlights the amazing complexity of this microscopic battle that's constantly going on within us.
It really makes you think, doesn't it? And it makes me wonder, considering all this, all these intricate interactions and the constant pressure of evolution and emerging diseases, what future breakthroughs in our understanding of these processes do you think are going to be absolutely critical for protecting human health?
That's a great question. I think a lot of the future breakthroughs are going to involve understanding these interactions at an even deeper level. Like, how can we disrupt specific virulence mechanisms without harming the beneficial microbes that live in and on us? And how can we harness the power of our own immune system and even our microbiome to fight off pathogens more effectively? There's still a lot of mysteries to uncover, but it's an exciting time to be working in this field because the potential for new discoveries is huge.
Very exciting and definitely a little bit daunting. Well, I think it's safe to say that we have comprehensively covered all the major points, theories, findings, and examples from our chapter on microbial infection and pathogenesis. We started at the very beginning and followed the journey of these microbes as they try to make us their home, exploring their strategies and our own defenses along the way. It's been a deep dive indeed.